So good evening. Really delighted to be here to welcome you to this tonight's Kavli Prize Laureate lecture. And tonight we're featuring Professor Bob Langer. We're honored once again, and we do this a couple times a year. We're honored to work with the Kavli Foundation, and you'll hear from its president in a few minutes, CEO in a few minutes, Cynthia Friend, the Norwegian Academy of Science and Letters, and the Norwegian Embassy here in the U.S. To join us to bring in these great scientists who have just recently won the Kavli Prize, which is, you'll hear, is a remarkable thing. Before we go into the conversation and we hear Professor Langer and Professor Frank Sesno, I'm going to invite Cynthia Friend, as I've already said, the CEO, the president of the Kavli Foundation. Cynthia.
Thank you, Eric. It's absolutely a pleasure to be here, and in particular to welcome Bob Langer from MIT, who's one of the 2024 Kavli Prize Laureates in nanoscience, and I'm happy to say a personal friend. The Kavli Foundation, which I've had the pleasure to lead since 2021 after a long career as a faculty member at Harvard, is a US-based philanthropy that supports fundamental science in 4 fields: astrophysics, nanoscience, neuroscience, and theoretical physics. And our founder, Fred Kavli, articulated a fantastic vision, which is to support and advance science for the benefit of humanity. And I have to say, I don't think it gets any better than that. Fred Kavli was a Norwegian-born American entrepreneur. He wanted to fund these scientific areas. He wanted to fund basic science in an enduring fashion. And in fact, the cornerstone of Kavli Foundation programs is a collection of 20 Kavli institutes worldwide. 7 of them are outside of the United States, which creates a network of scientists and scientific research in our fields. Currently, the Kavli Foundation, under my leadership, is also supporting emerging areas of science that have potential for transformative impact. And we'll hear about some very transformative work that Bob Langer has done in his research career. Now, Fred Kavli also understood that it was very important to make strong connections between science and society, because we rely on taxpayer dollars to support science, but also it's important for everyone in our culture and our society to understand something about science. And so to that end, he actually created the Kavli Prize, and it's a wonderful partnership, as Eric alluded to, with the Norwegian Academy of Science and Letters, and also the Norwegian Ministry of Research and Higher Education in Norway. And so they are partners with the Kavli Foundation. And as you'll hear in a moment, the Norwegian Academy of Sciences and Letters actually administers and takes care of the selection process. And I have to say they do a wonderful job. We support fundamental science at the Kavli Foundation because we believe in its importance, the strong impact that fundamental science can have on our future. The science and technology has had all kinds of impacts that we couldn't have imagined when those discoveries were initially made many years ago. And in fact, I think that applies considerably to what we're going to hear about from Professor Langer today. The work that he's done with integrating nanomaterials with biomedical devices has been absolutely transformative and incredible. And of course, one of the things that you will be familiar with are the Moderna vaccines with the mRNA, which has been really pivotal in helping us control the COVID-19 virus. And Bob actually was a founding member of that. He's really a remarkable visionary, and I have to say he's the only person I know on this that actually is honored in a kind of a funny way, perhaps, but at Logan International Airport in Boston, there's this long timeline that spans, I think, a couple of hundred years, where they cite various innovations and inventions that were really important to the world. And Bob is actually featured on that. So if you go to Boston sometime, go to Terminal B and you'll be able to see that, maybe elsewhere as well. So the Kavli Prize has been awarded since 2008. There are 73 laureates overall, and 8 were awarded this year. And we're really pleased to have Bob Langer be sharing with us the work that he's been doing. And I want to take this opportunity now to actually thank Frank Sesno, who is the moderator of this event, and he does a wonderful job of facilitating these conversations. Before I move on, I also want to thank Eric Isaacs, the president of the Carnegie Institute for Sciences, but also the Carnegie overall for working with us on this event. So since 2009, the Carnegie Institute has collaborated with the Kavli Foundation to have these science evenings through the Capital Science evenings that the Institute, that Carnegie Institute has established. I think it's really created a very loyal following of people interested in science. And so I'm really glad that all of you are here braving the weather tonight. So thank you very much. We value the partnership and we look forward to continuing it for many years to come. So finally, I just want to ask you to please join me in listening to a recorded message from President Lisa Overås. She's the president of the Norwegian Academy of Sciences and Letters. She unfortunately couldn't make the long trip here for this occasion, but she did create some video remarks, which will be shown now. Thank you very much.
Good evening. My name is Lisa Overås, and I'm the president of the Norwegian Academy of Science and Letters, greeting you today from beautiful Bergen at the west coast of Norway. I really would have loved to join you in Washington today, but the journey is sadly too long to fit into my schedule this week. First, I want to express how much we in the Academy value that the Carnegie Institution of Science and President Eric Isaacs are hosting the Kavli Prize Laureate conversation again. Events like this are invaluable contributions to communicate the great achievements of the Kavli Prize laureates. Fred Kavli's vision for the Kavli Prize in the US and international is to recognize outstanding scientific research in the field of nanoscience, neuroscience, and astrophysics, to honor highly creative scientists, promote public understanding of scientists and their work, and foster international cooperation among scientists. The partnership to administering the Kavli Prize was established in 2005 between the Kavli Foundation, the Norwegian Ministry of Education and Research, and the Norwegian Academy of Science and Letters. The cooperation has been fruitful and remains key to the success and the international standing of the Kavli Prize. We also very much appreciate the ongoing support of the Norwegian Embassy in Washington, D.C. A few weeks ago, I had the pleasure of getting to know Robert Langer, Kavli Prize Laureate in Nanoscience, and tonight's guest of honor, when he and the 7 other laureates of 2024 came to Oslo to receive the Kavli Prize from His Majesty King Harald V. Robert Langer is one of the most prolific and successful scientists of our time. His research on nanomaterials for medical purposes has revolutionized the pharmaceutical industry and saved millions of lives. In meetings with Norwegian emerging talents and the press, Langer chose to emphasize how fortunate he has been and the importance of simply being a nice person in science. His admirable combination of excellent skills, dedication, and humility makes him a brilliant role model for new generations of scientists. And hundreds of them has had their training under his mentorship at the Langer Lab. I'm so much looking forward to listening to the recording of Robert Langer's conversation with the eminent Frank Sesno, and wish you all an inspiring evening. Thank you so much for your attention.
Okay. Professor Langer will have to, please come up. We'll have to suffer a little bit more aggrandizement here. Thank you, Cynthia, really, and thank the Kavli Foundation. We love this partnership with you, with the Norwegian Academy of Science and Letters, and with the Norwegian Academy Embassy here. So it is a really rare privilege to get to introduce someone of Professor Langer's stature. Actually, it's not rare, it's unique. It's really unique. And you'll see why in a minute. I'm just going to give a few numbers and then you can have fun with the discussion. But Professor Langer is the David H. Koch Professor, Institute Professor at MIT in the Department of Biological Engineering. But the reason we're here tonight is because Professor Langer also won the 2024 Kavli Prize for Nanoscience, which you've heard, alongside two other remarkable scientists, Professor Chad Merkin at Northwestern, and Professor Paul Alvisados, currently president at the University of Chicago. I just want to read the citation because no one's quite done that yet. So if that's okay. The citation that Kavli was, for all three of them, was combined nanostructure synthetic materials with biologically active molecules. They have pioneered their use in therapeutics, vaccines, bioimaging, and diagnostics, contributing foundationally to the field of nanomedicine. In addition, the three laureates have contributed to the societal impact of their research by founding companies to translate their fundamental science into biomedical applications. And that is really what makes this award very special. This year, many years, but this year in particular, nanoscience maybe 20 years ago was kind of becoming a reality where little particles, little things you'll hear about today were science, really interesting science. I was involved in it myself. But what Professor Langer has done is he's taken nanoscience and he's translated it into some remarkable discoveries in science, but also taken these discoveries, as we heard, to really help humankind, which is the remarkable part. He has just a couple numbers. I have to say this. In the Google Scholar has said that he is the most widely cited engineer in history. He has 1,600 publications. Any of you, a scientist here, has got 1,600 publications, and 1,400 patents. So you can ask where he gets the time, but that's another thing. And as we've already heard, some of those patents have gone on to really make a difference. You may remember the company that's still very active called Moderna. Okay, I don't need to say any more about that. Really remarkable. And I will say one last thing, that he's also been prolific in educating students. In fact, this just a few minutes ago, we had the privilege of meeting one of his students who is a great scientist, but now she wants to go into looking at policy and politics. So it's a great impact that he's had with basically 400 alumni and 500 who have worked throughout the world. So we're really excited to hear what he has to say. I won't go on and on with the numbers, but Professor Langer is truly a giant among scientists. Now, we also have a distinct privilege of having Professor Frank Sesno here. We know Frank very well. He's worked with us many times. Frank is a professor at the George Washington University School of Media and Public Affairs, and he also serves as the executive director of the GW Alliance for a Sustainable Future. So he's doing great work too. You may know him, you may recognize his voice. Prior to his work at GW, he spent 20 years at CNN. He was a White House correspondent. He was the bureau chief here in DC. So even before I met you, Frank, I knew your voice. So it's great. And he's also been a great friend, as I said, to us, to Kavli, and done a spectacular job, not just talking about science, but pulling the person out of the scientist. And so with that, I'm going to now turn this over to Frank and to Bob, and look forward to a really fascinating conversation. Thank you.
Thank you very much, Eric and Cynthia, and to all of you who've joined us this evening, and those of you who are joining us online. Welcome, welcome. And Bob, what a great pleasure and privilege to be with you again.
Well, the privilege is mine. I'm very, very happy to be here. It's a great honor and certainly a great place.
There is so much for us to talk about, and I'm looking forward to your questions, which you can put up via Pigeonhole, and we'll go to that later. I was struck by the data points, right? 1,600 publications, 1,400 patents. There's another number that I just learned. You're a big walker, right?
Yes.
How much do you walk?
I try to walk over 10 miles a day. I guess I've done that for a while. I used to run, but now I'm getting old, so I hurt my back, so I do more walking.
How do you have the time to walk 10 miles a day and do 1,600 publications at 14?
Well, the publications, what's important to realize is I have great students and postdocs, and they keep coming to my office with great papers and patents. So that's kind of the easy part. I mean, it's evolved over time. I've been doing this for 50 years, not the walking, but I've done that for a long time too.
Well, we have a lot we'll cover tonight: your science, your story, how you started. Let's start with the science. And for those, we hear the term nano and nanoscience a lot. For people who know it, they probably know it. but others may hear it and not be so sure about it. So what are we talking about here, and how small is small? Just remind.
Yeah, well, I suppose a classical definition is to say that you're studying something, that might have a diameter between, 1 and 100 nanometers or 1 and 1,000 nanometers. But another way to think about it is it's maybe on the order of 1/1000th the thickness of a human hair. So it's very tiny.
And one of your big breakthroughs, as you did this, was this sort of dramatic, hard-fought hypothesis, assertion that material could be nano-engineered. Explain that.
Well, I mean, in our case, nano-engineered means that you do something with it that really wasn't done before and engineer it for a particular purpose. In our case, what we nano-engineered these things for is to take polymers and lipids, make them really small, but use them to encapsulate molecules, drugs, and particular large molecular weight drugs, which people, when I first started to do this, didn't think was possible.
As Eric mentioned when he was reading the Kavli citation, your breakthroughs have combined nanostructured synthetic materials with biologically active molecules. So that is combining and doing what? Yeah,
well, the big area that we studied is what we'd call drug delivery. Not like a mailman or something like that, but basically that you could take something, take the pharmacologically active molecule. I might as well use the one that was mentioned because, even though it wasn't exactly what I did 50 years ago, but a great example is if anybody's had the COVID vaccine, what's done is to take, in this case, messenger RNA, which is a big molecule. That's all what we were doing. There's a paper we wrote 48 years ago where we showed you could take very big molecules, actually DNA and other nucleic acids were among them, put it in a very, very tiny particle, and then show that it could come out and you could deliver it to the body and get a biologic effect. So in the case of the COVID vaccine, the biologic effect is that hopefully you don't get COVID as frequently, and maybe if you do get it, it's not as bad, and that's because you gave it, and it's a whole new way of taking vaccines.
We have a video here, an animation that sort of illustrates this a little bit. Let's take a look, and then you can tell us what's happening.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So in this particular case, obviously, cancer.
Yeah.
What's happening?
Yeah. So actually, and so this, just to give the appropriate credit. So Nova, the TV show, on PBS, they came to our lab and they filmed a lot of things with me in it, and actually they did an amazing job. They also made that video. And this must have been around 2009 when they came. So obviously COVID wasn't around and none of these things, but we had already created nanoparticles that you could put different molecules in. Also, part of what they're saying, the first challenge is to be able to make it and have it come out. Another challenge, though, is when you make the nanoparticles, they can stick together because it's tremendously small. It's like much smaller even than wet sand, which would stick together. And also, there are cells in your body called macrophages can tend to eat it. So what we were trying to do, and still trying to do, but we've had some success, is come up with cancer treatments where you could put a drug, maybe a cancer-killing drug, in a nanoparticle. You really can put anything in it now, and put a target, put something called polyethylene glycol, or PEG. That's that coating that they showed. But then maybe even have a targeting molecule that would hopefully take it to the cancer cell. Now, of course, it doesn't really blow it up. I always say all the clinicians I work with tell me it
took a little creative license there.
Yes. Oh, yeah, they did on the show, but they did a great job. I mean, they had 6 minutes of different things that we'd done, and but basically, the idea is that you could create this nanoparticle of these different components and then have it target a cell. And even though cancers earlier, there are good examples of using nanoparticles with what are called siRNA to treat a rare disease. That's actually a nerve disease called ATTR amyloidosis, and it's helped many, many people.
And this is the technology, and as you're explaining, these drug delivery systems, these controlled drug delivery systems that have contributed to incredible therapeutics, vaccines that we've seen, we were just talking about that, bioimaging, diagnostics. Where is this most promising right now?
Well, I'd like to think that where it maybe is going to be most promising, though I'm prejudiced, is in nucleic acid delivery. So messenger RNA is a great example. I mean, just to pick an example, Moderna and Merck are in advanced clinical trials for new cancer treatments, and they're using it to make what's called an individualized neoepitope or neoantigen therapy. But another people might call it a personalized cancer vaccine. But what you can do is, let's say, I'll just give the real example. If somebody has melanoma, but it could be any cancer, but in the melanoma trial, what they did is they took the person's cancer cells and they analyzed it, and they found for each person what were the 34 most common what are called neoepitopes. That these are mutations that make you have cancer. And then what we did in the case of COVID, you make the vaccine against what's called the spike protein. That's in COVID. Here, you're making it against these 34 neoepitopes. So the idea is that you give that to the patient, and the hope is that and the beauty of messenger RNA is you can make these quickly before there's too many more mutations. So the idea is you give it to the patient, and basically the body will start making all these cells and also antibodies, hopefully, that will go against the existing tumor and destroy it. And in the clinical data, what was done, this was a double-blind trial. It's so far been done on like 157 patients. Merck has a what's called an immunotherapy drug called Keytruda, which is probably really one of the best drugs around for treating cancer. So what they did is half the patients get Keytruda, half the patients get Keytruda plus this vaccine, and you don't know who gets which. But basically what they were able to show is after 2 years, you got like 44% of the patients that got the Keytruda plus the vaccine had a much lower, didn't have a recurrence of the cancer or death compared to the
44%.
44%. 3 years, it's 49%, and metastasis was reduced by 69%.
Wow.
So I mean, it's still what's called a phase 2 trial.
Sure.
Now they're doing a phase 3 trial, but that's an example. And of course, the hope is that it'll be able to be used for other cancers. It'll be able to use for other vaccines. It'll also be able to be used for all kinds of rare diseases to make enzymes or something where people may not have an enzyme and have an enzyme deficiency disease.
One of the amazing things about the mRNA that we had for the vaccines for COVID is how rapidly that was developed, right?
Yeah.
History making. Same thing going to happen with these cancer treatments, you think?
Well, yes and no. So there's two things that made the vaccine fast. One is the technology. And in that case, that's the same. The other, of course, are regulatory authorities. And that's not the same. I mean, I think here with people dying right and left, the regulatory authorities really move quickly. And so, but, the FDA's policy is basically like the Hippocratic Oath, first do no harm. So they want to make sure that it's very safe too. And that you do larger numbers. So the classic way of doing these things is a phase 1 trial. You wait for the data for that to find the highest safe dose. Then you do a phase 2 trial to see what that dose does. And then the phase 3 trial, which is very large population. So they want the companies to do that.
So I'm a Moderna guy, maybe a few Moderna people in the room. I want to put another slide up because this has a story to tell too that I want you to tell us. This is from Forbes from 2023, right? We're looking at vaccines, 49 failed, 2 approved, 209 failed treatments, 1 approved. antivirals, 153 failed, one approved. What's the story here?
Well, the story here is a lot of people tried, right? I think whether it was in China or Europe, and really the only two approved is the, for a long time now, Novavax has just gotten approved, but for the longest time, people tried all kinds of conventional ways to do it, and they didn't really work very well. By the way, I think an important reference point is flu vaccine. Flu vaccine is like 40 to 50% effective, and they're guessing, when they make flu vaccine, you have to grow the eggs up well over a year in advance. You don't know whether you're making H1N1, H1N2, so you guess. The reason it's so ineffective is because you are guessing and you're going to guess wrong. Here, you knew exactly what you're going after, and so you could do it much more rapidly if you use the messenger RNA approach. So basically, there's two companies, one in the United States, which was Moderna, the other in Germany, which was BioNTech. Pfizer in 2020 made an agreement with them that they would help them and distribute it and help in manufacturing. So those are the only two. They're both nanoparticles with messenger RNA. None of the other approaches worked.
But these are yours, these successes, right?
Yes, well, they're based, all of it.
Based on your...
Well, the earliest studies that we did, there's a number of contributions we'd make over the years on this. One was the idea that you could create nanoparticles or microparticles that could deliver large molecules. And people, when we first did that, didn't think it was possible. But then we did other things, like I mentioned, the PEG coatings, we were involved in that. We also created other components that would end up being used in lipid nanoparticles. And we even developed, again, these are different students, Rohit Karnik in this latter case, and Omi Farrokhzad, developing ways of even manufacturing these early stage, which Moderna and BioNTech and Pfizer all amplified tremendously.
Can I just ask you, as a human being, as a scientist, what must that feel like to have contributed to something like that when the entire planet is suffering and dying, and you help develop a response?
Yeah, well, it was, I would say, I was on the board of Moderna when they broke the code, and it was, it was pretty emotional experience for everybody there, including me. You don't know what the results are when the trial's double blind. So when they showed us the results, it was a remarkable moment.
What was that like?
Well, it was exhilarating on one hand. I mean, I think I wasn't sure what the results were going to be. So, you never are when they're done that way. But I've always believed in the technology, and I was certainly hopeful that it would happen. I've always done this kind of work, not just because I like to do curiosity-driven research, which I do, but I also want to see it make a difference in the world. And that meant a lot to me. The other things we'd done before that, some of them have also made a difference, but this was like a striking thing.
Yeah, I'd say so. Aggressive brain cancer, prostate cancer, schizophrenia are among the kinds of things that may be addressed by some of these discoveries. And I'm wondering if you could explain why these particular medical conditions may be responsive to this kind of treatment.
Yeah, well, there's different variations of what we've done. I mean, the nanoparticles, and I think it's really important to realize that it's not just me. I mean, we got things maybe started. There are thousands of people. Our students are among them, companies. I mean, people all over the world have, I think, made enormous contributions. But so you can use them in lots of different ways, right? You can make a lipid nanoparticle that you can inject that might be useful for the vaccine. You can also make a polymer one. You can inject these underneath the skin and make it last actually for a really long time. So some of the ones that you mentioned, you can't, people can't take orally. If they try to inject them into the body, they're just destroyed right away. So you're protecting them. That's also what happened with messenger RNA vaccine. It's protected. I mean, you could give messenger RNA, but it's destroyed. So you could take proteins, you could take small molecules, and you could make them last much, much longer than you normally could. And that does two things. One, you don't have to inject yourself in some cases every minute or two, which wouldn't be possible. And and two is that there's a problem a lot of times with what's called compliance. I think the New York Times had an article a few years ago based on a medical journal, Annals of Internal Medicine, that something like, millions of people just don't take their drugs. It causes enormous number of deaths, hospitalizations, and so forth. And that's particularly prevalent in certain diseases like cancer, like schizophrenia, opioid addiction, and so forth. So,
yeah, I'm particularly interested in the schizophrenia. How would this address that?
well, the biggest one of the issues in schizophrenia, I mean, I don't know if people saw A Beautiful Mind, the movie, but probably a lot of people did. So that's an example of schizophrenia, and Russell Crowe got electroshock treatment in that case. Now they're drugs, but again, if you're depending on the person, if people, a lot of people just don't want to they don't want to take it. Either they don't want to, or they forget, or they're in a state where they just don't do it. So now you can give it injections once every couple of weeks, once a month, all based on permutations of the type of thing we just said.
Let's talk about your journey now,
Sure.
because this is a fascinating journey. When you started, this was a big uphill thing for you. In fact, you've said your career has not been straightforward, sort of followed the Robert Frost road not taken
Right.
path. A chemical engineer, you went from chemical engineering to nanotechnology to biology and medicine. And you say along the way you experienced, these are your words, rejection and ridicule.
Yeah. Right.
Rejection and ridicule?
Yeah. Should I go through it all?
Well, maybe not all of it, but
Yeah. Well, I mean,
it's a great story of persistence, right?
Sure. Well, so again, just to go over my early career. So I got my degree at MIT, which is a pretty good place, my doctorate in chemical engineering in 1974. And, when most of and if any of you remember that, and if you were in Boston, there was this gas shortage sort of, and if you had a car, you actually had to wait in line for 2 hours to get your tank filled up. But the consequence of that is that if you were a chemical engineer, you got a lot of job offers. And pretty much all my classmates that year went to work for oil companies, and I thought I'd do that also. So, because that's what my friends did. So I actually got 20 job offers, 4 from Exxon alone, and I wasn't that good. But I remember flying back to Boston thinking to myself, when they were talking to me at these companies, a lot of it was like increasing the yield of certain petrochemicals by very, very tiny percentages. And I just felt I didn't want to spend my life doing that. So I started to make me think, well, what did I want to do? So one day I saw an ad to be assistant professor at City College of New York to create new chemistry curriculum. And I was particularly excited about that because when I was a graduate student and I was supposed to be doing my thesis, I spent a lot of time helping start a school called the Group School with others for poor kids. And I got very involved in trying to make math interesting and chemistry interesting, at least I thought I was. And so I was, we created a whole book on, on chemistry experiments. So when I saw that ad, I wrote them a letter applying for a job to be assistant professor to create new chemistry curriculum, but they didn't write me back. So, but I liked the idea. So I found all the ads I could to do something like that. I think I found about 40. I wrote, and they weren't necessarily great colleges, but I was excited about this idea. So I wrote them, and I don't think any of them wrote me back.
Wrote 40
Yeah.
applications and
Yeah,
crickets from
I didn't do very well. So at any rate, but that still got me thinking, like, what other way could I use my chemical engineering background to help people? So the next idea I had was medicine, which wasn't really done much in chemical engineering then. So anyhow, I wrote to a lot of hospitals and medical schools, and none of them wrote me back either. But the thing that happened then was I walked into the lab one day, and one of the older scientists said, Bob, he said, there's a surgeon in Boston named Judah Folkman, and he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, so I went to work at Children's Hospital, and I was the only engineer there. Anyhow, so that was a start. I could go over more, but.
And how'd you do on those early grant applications you were writing?
Yeah, well, so a couple of things that are important. So I did terrible in those, but I have to explain why. So, you know, the problem that I began working on in Dr. Folkman's lab, he had this idea, was that if you could stop blood vessels, you could maybe stop cancer. And I thought it sounded amazing. But it turns out he was widely criticized, and people didn't believe it. So he hired me to isolate what might be the first blood vessel inhibitors. But to do that, I had we had to create bioassays. There was no way to study blood vessels. That's where some of the nanoparticle work came in and microparticle work came in. So anyhow, we published papers eventually in Nature and Science, which are good journals. One on the fact that for the first time you could take tiny particles and release large molecules, which these angiogenesis inhibitors were, and two, that you could use it actually for the first time to isolate substances that could stop blood vessels from growing. So, but the first time I actually gave a talk on this, I remember like the audience came up, I was 27 years old, and there's a very senior audience in Michigan, and a whole bunch of the senior scientists told me they didn't believe anything I just said. So that wasn't so good. And then as Frank was just saying about my grants, so I tried to get grants for these things, and my first 9 were rejected. What happened was that on those 9 grants, this is my interpretation, but you know, they'd go to NIH study sections or things like that, like pathology B study section, and they would write things like, well, Dr. Langer is an engineer. He knows nothing about cancer and even less about biology. Anyhow, so I didn't get any for a long time. And then the other kind of rejection, I love being at Children's Hospital. I really learned so much. And Dr. Folkman was a wonderful person to work with. But a lot of my friends after 3 years said, you know, you should probably get a real job. And so, you know, I thought, well, maybe they're right. And I was starting to get some ideas about what I could do. So I wrote to, I'm a chemical engineer. So I wrote to chemical engineering faculty jobs, and no chemical engineering department in the world would hire me. They sort of thought that, this is, again, my interpretation. Maybe they just thought I was dumb, but but basically my interpretation was that, they thought I should be doing like mathematical modeling or energy or oil or something like that, rather than experimental applications of, I think, engineering and chemistry to biology. So I didn't get any jobs there. Finally, I got a job in a nutrition department.
Is that what made the difference? I mean, when did success, what you would define as success, start happening?
Yeah, well, that's a very hard question because it gets to that definition of success. I thought
being taken credibly,
yeah,
being not ridiculed.
Yeah. Well, it was a slow process,
Yeah.
I mean, really it's certainly, for the most part, I think he, at least, Nevin Scrimshaw, he was the head of the nutrition department. He thought I was credible, so that's why he hired me.
Did you get discouraged along the way?
Sure.
Did you get like, I'm just going to go be an airplane pilot or something like that?
Work in an ice cream parlor because I like I like ice cream. Nobody would want me flying an airplane, but I definitely
I figured engineer,
Yeah, well, but I've always liked ice cream, so I did think about things like that.
Seriously?
Yeah, sure. I mean,
Could have been Ben and Bob's instead of Ben and Jerry. Right?
Well, there's yes. I could tell you there's a Ben and Bill, so the candy shop. But at any rate, so I would see over time. Really, what happened initially is when I was in the nutrition department, the Nevins, even though he hired me, he left after the next year, so that was not so good. So a lot of the senior faculty wanted me to leave. But I found that over time, what happened, I mean, this is how I think MIT works. They write letters to people, right? And even though there was a lot of internal people that were not necessarily for me, when they wrote to different people, some other professors and at different colleges, and actually the president of Merck, they all said, boy, this work is important.
I want to ask you what you're proudest of, but I want to put up another slide because this is something. In 2014, from Professor Michael Marletta, UC Berkeley, member of the National Academy of Sciences. He wrote, and you talked about this. One evening, I went to a faculty dinner at a Chinese restaurant with Bob Langer and some senior MIT professors. A senior scientist sat quizzing us while smoking a cigar. When the older scientist heard Langer's concepts for drug delivery, he blew a cloud of smoke in Langer's face and said, "You better start looking for another job." Maybe that's when you were looking for the ice cream parlor. I thought I was in a Fellini movie, he said.
That's correct.
Now you're one of 9 institute professors at MIT. It's the highest honor that can be awarded to a faculty member. We've heard about your remarkable successes and patents, businesses. You joined, MIT back in 1978. When you think about this remarkable, I mean, it's really an unbelievable career that you could never have, I'm sure, in your wildest dreams,
Sure.
thought about in those early difficult days. What are you proudest of?
Well, really, when I mean, this may not be, but really, though, what I'm proudest of is how well my students have done. I'm certainly proud of the science, but I feel when I look at how well my students have done, I mean, they've won all kinds of awards. I think 26 are in the National Academy of Medicine, 23 in the National Academy of Engineering, 3 in National Academy of Sciences, I think 48 in the National Academy of Inventors. And then they've won the Waterman Award, they've won the National Medal of Technology. I mean, all kinds of things. And I'm not to be snobby about schools, but I think 10 are professors at MIT, 11 at Harvard, and 12 at Stanford. And so, and there were professors all over the world. I mean, so and they've started great companies. So, I mean, I'm very proud of that.
As you should be. Let's put up this next. Nothing more rewarding than seeing your students succeed, and you have seen unbelievable success. And they like you too. I mean, they helped you celebrate your 70th. We have a photo to put up here. They help you celebrate your 70th, and you have sort of an annual gathering.
Yeah.
And they treat you with great seriousness and respect, right?
Right.
Yeah. Little funny thing along the way.
Well, I mean, if I go to that particular picture is actually from last year at our lab party, and people bring kids and spouses, and that's me and my wife and our granddaughter. It was circled. But I'll go back to the 70th, where it was kind of a reunion, and so a lot of people came from back even from the 70s who were working in our lab, and I think over 700 people came, and they spent about 3 days they'd give scientific kind of lectures, but they also told a lot of stories about me, so which were funny, and but it was a pretty remarkable thing. Yeah.
That's great. It's great. I want to put up another slide, really. Engineered tissues. That gives us a sense of what's going on. Where's the field going, Bob?
Well, I mean, so this is more broadly on bioengineering and the intersection of the intersection of engineering and biology, or just biomedical engineering. Not just nanotechnology, but nanotechnology is certainly a part of it. I mean, drug delivery, which we're doing. I mean, there's still so much more that needs to be done. You hear about things like CRISPR, and could you do that in what's called in vivo in people, which is a big delivery problem. Can you target nanoparticles to cancer cells or other cells? So we're working on a lot of those kinds of things. But there's so many other aspects of material science, whether it's sometimes nano or sometimes other things, combined with biology. So one of the other big areas that we did a lot to start, along with Jay Vacanti, who was a surgeon who came to see me back in 1981, was, could you make tissues and organs from scratch? That's what that slide shows. That's an area that people call regenerative medicine, and many people have contributed to that too. But I mean, I could go through the whole slide, but basically, you can now make. I think if you look on the top left,
Skin?
Top right, yeah, you can make skin. You can see I can't even figure out my right from my left, but
we'll let that go.
Yeah. But anyhow, you see, you can now make skin for burn victims. I mean, I have full slides of this, but I have this composite that I make at the end of some talks just to give a feeling for where it's going. But the idea is that you could someday make almost any tissue or organ. Another example up there, this is done by Laura Nicholson, who was one of my former fellows, and she then became a professor at Yale, but then started this company, Humacyte, which to make new blood vessels, and which started when she was with us in the 90s. She 1999 paper in Science. And that actually is at the FDA right now. It's finished the phase 3 trials, but it's already being used, like, you know, for soldiers in the Ukraine who have been badly wounded, and to make new blood vessels for them. But theoretically, and I mean that because it may take a long time, it will take a long time, you might be able to make a new pancreas for people who are diabetic, help people who are paralyzed by spinal cord repair, help hearing. But the other big thing that it enables, I think it is enabling, is that you could think about making organs and tissues on a chip. And you see that with the brain, with the gastrointestinal tract, and the heart up there. Those actually are all taking cells from people or animals and growing them outside the body, but imparting the same characteristics we'd like to think that are inside the body. And there are tests that you can do to do that. You might ask, well, why is that important? Well, one is someday it could tremendously reduce animal or human testing if you could test something on things like this. In fact, the FDA has been particularly interested in that. Two, it could tremendously speed up drug discovery. Right now, when you try to do studies on drugs, I mean, you know, people have been working for 40 years on Alzheimer's disease, you know, and they still don't understand it very well. But if you have something here, like in almost a test tube, like, you could do literally thousands of experiments. So it could tremendously speed up drug discovery. In fact, that brain on a chip, that's been done in collaboration with Li Wei Sai at MIT, who's head of MIT's Picower Institute. And I've had this great postdoc from Stanford, Alice Stanton, who's led a lot of that. So again, I don't want to make it sound like these are done. I mean, there's still so much more to do, but that's another example. And there are many other examples too, I think, of where engineering and materials will be able to help people. I mean, in fact, in the vaccine area, we've been working on what we call, in fact, this came from Bill Gates. He once visited me, and they fund a lot of things in our lab to help develop the people in the developing world. But one of the questions he asked was, could you create what's called a self-boosting vaccine? Just give a vaccine you could give once, and yet it would deliver doses at different times because of the way we engineered the materials. You could think of tiny eggshells, and the vaccine is in different eggshells, and we can make those little eggshells, like the nano eggshells, break at 1 month, 2 months, 1 year. And we're also creating like microneedle patches that could maybe be shipped all over the world to give them. And there's so many other areas too that bioengineers are working on. You know, I mean, like Chad and well, they're chemists more than chemical engineers, but they've come up with new imaging techniques, new types of DNA that could be used for helping molecules cross the blood-brain barrier. And there's so many things going on for the future.
Incredibly exciting, fascinating stuff. I mean, it really is amazing. A lot of questions from the audience. So why don't we dive in and see what we can do? Some really interesting ones here. And thank you for the questions. First, how can chemical engineering help address the addiction crisis?
Well, so that actually is a picture. I didn't go through that. It's already doing it, I think. So you can, not that it's solved by any means, but there are molecules that make you, that make you naloxone and things like that may help, but people don't take them. So one of the things that's already being done is a once-a-month injection of that. That was one of the microspheres up there, and that's, I don't want to try to say it solves it, but it does.
Been successful, more successful?
Yeah. Oh, absolutely. It certainly. I mean, and lots of people, I think, do use it. And there's other things like that.
And is that growing? Is there?
That's, yes. I mean, it is. As far as I can see, Alchemies is a company. I was actually on their board for 10 years. They acquired a company that we started And yeah, I mean, that's according to when I look at the sales, they does keep growing. Yeah.
Next question. How will AI and quantum computing likely change the fields of therapeutic macromolecule discovery and delivery?
Well, so AI, I mean, well, quantum computing, I mean, I don't know when that will necessarily happen. I mean, from people I talk to, but when it does, it'll certainly change a lot of things because you'll get so much, you can do so much. But AI already, you know, what's happened is there are things like you've probably seen, or some of you people may know of, like AlphaFold, you know, done by Google's DeepMind. And so you can make really maybe make better antibodies, which is a kind of you know, that's a lot of drugs are antibodies now, and those are also large molecules. So you know, you could so that's but AI, you have to have a lot of information to make, you know, like a lot of data to really make AI useful. By the way, AI also, you know, like Regina Bargeli at MIT, they're looking at for better imaging and analysis, like for breast cancer, as an example. But so I think AI will help. I think it'll help a lot of things. There's a lot of things that won't help that people are saying it will help, but
You think it's going to be like this magic moment transformational thing?
no, I don't think any. And by the way, AI is not new. I mean, not to brag about MIT, but a lot of it started 70 years ago, and so it's really not new at all. But it keeps getting, you know, it's computation, and that's the point about quantum computing, is that keeps getting more powerful. The things you can do with it, of course, become more powerful. What happened, in my opinion, is all of a sudden, a couple of years ago, you got this dramatic demonstration of what it can do with ChatGPT.
Yeah.
So now all the newspapers and everybody think that it's brand new, but it isn't.
And some students.
Yes, yes.
It's
but it has been continued to evolve for, like I say, a good 70 years.
Sure.
And will continue to do so and get better and better.
Next question. To what extent, this is back to COVID again, to what extent was the federal government's Warp Speed program helpful to the vaccine effort? I think you talked about that a little bit, but and what, if any, lessons should be learned for the next similar effort?
Well, I think it was incredibly helpful. I mean, I don't think when we started it, we knew that, I mean, a lot of people, like, I didn't really go over this, but, you know, a lot of people thought Moderna and BioNTech, you know, these two tiny companies, they thought they were never going to work. Boston Globe, I think, our hometown newspaper, even after we had the phase 1 data, had a front page article with my picture there saying this is not how you do science. So there's a lot of skeptics.
They're still after you, aren't they?
They are. Yeah, but and probably always will be. Anytime you try, no matter who you are, I think unconventional ideas, you know, that's, people are going to criticize you. But that being said, you know, the fact is that it gave funding for multiple shots on goal. I think that was, you know, really helpful. It's kind of like David Koch, who gave a lot of money to MIT. One day he was talking to me about horse racing, and he said, Bob, he said, I figured out how to always win the Kentucky Derby. And I said, what'd you do? He said, well, I just bet on every horse. and
some people can do that.
Yes, well, he could do that. So at any rate, that's kind of what, I mean, you didn't bet on every horse, but it helped enormously. Could you duplicate it? Well, I mean, if you put a lot more money into something, I don't know that you duplicate it, but you would make it go faster.
Bob, here's a question that is something that I work a lot with and care a lot about, and I'm very concerned about. So it's a very topical, relevant, urgent question. How can we help combat misinformation about the pharmaceutical and vaccine industries?
Yeah, well, I thought you were going to ask something about misinformation, and I don't. I mean, I think that's really a hard thing to do. I wish you could. You know, I keep thinking about it myself. And I think the misinformation is particularly prevalent in the United States. You know, there was an article that I read which talked about how, you know, and maybe that wasn't their intent, but just as an example, how I think it was one of the national places that wrote this, that something like in the US in the year 2020, 90%, over 90% of the articles on COVID and the vaccines were negative, whereas in Europe it was about 50-50. And why is that? I think some of it's political, I think. And I also think, though, in the United States, my wife mentioned to me, and I don't know the exact numbers, something like 30% of the people in the United States don't believe in evolution, and I don't know how it all goes together, but I think that education to me has got to help some, but I think probably so I think really giving kids at every level good education, that'll help some.
I mean,
but
we appear to be backsliding in a lot of areas on the vaccine front, whether it's childhood vaccines, flu vaccines, COVID vaccines,
right?
The booster, people are not getting the boosters as they should be and it's a whole information ecosystem.
I agree with you. Now. Yeah, no, I agree with you. I don't know the answer to it. I think the media could help, but you have all these different types of media. I'm sure I don't have an answer on that better than anybody else. I wish we did. I mean, but
it's one of the things we've talked about too, is how can we help science and scientists tell their story better, and connect with people in more ways and more places. A couple more, and then we're going to be out of time here. I love this one. Thank you for the mRNA, little red heart here. What keeps you going now?
Well, I think the same thing that's always has been. I mean, first, I love doing research. I love working with students, but I also love seeing us have an impact on the world in what I hope is a positive way. So I mean, it's all those things.
So here's a perfect follow-up. As an early career researcher, somebody's an early career researcher, is it important to focus on applied results, or is it okay to just pursue curiosity for curiosity's sake?
Either. I mean, people should do what they like, and maybe both. I mean, I think pursuing things for curiosity's sake is wonderful. And I mean, in fact, I got what I started doing in the beginning really had nothing to do with a lot of the things we did later. It was just to try to figure out a way to understand how blood vessels were growing and how to stop them, and that was certainly curiosity. But I think once you do something, at least to me, I feel like I wanted to see more happen than just write a scientific paper, but I think either of those, basic curiosity-driven research is fine, and applied things are fine. It's a question of what's their passion.
I want to close on this before handing it back over to Eric. We heard Lisa Ovas from the Norwegian Academy say in her comments when we started, she noted your emphasis on the importance of simply being a nice person in science. Mm-hmm. What's that mean?
Well, I guess, I mean, I hope people are nice, but I did learn lessons, as running a lab. One time I had somebody that was not so nice not to me, she was fine, but she wasn't nice to some other people. And that really creates enormous difficulty in getting scientists to work together in a small lab if one person isn't so nice. And I mean, I think having nice people work together, the environment is such that, I just think you have a collegial environment where everybody's trying to help each other. That to me means a lot. And I think we do better when everybody's nice. Plus, I think this is going to sound trite, but I think the golden rule is a pretty good rule, about doing unto others as you'd want them to do unto you. I mean, who wants to be treated badly?
Okay. I think your track record shows that you have been nice, but much more than that. And you have helped to educate and motivate multiple generations of scientists who are like you, making contributions, changing the world, advancing humanity. And I don't think we can begin to thank you for all that you have done. So, Bob, thank you.
Thank you. You did a great job. Thank you.
So I just can't help myself to come back to the second to last question, which is I think what we've seen tonight is the most stellar example of the fact that there isn't just basic research and applied research. It's absolute continuum. And you have somebody who's excited by basic science, creating these little shells that, nanoparticle shells that can do stuff, but then also figuring out how to use them to deliver drugs. It's just an amazing continuum. So fundamental science is important, applied science is important, but most importantly, the funding for this stuff. and I really, it's not trite. We're here in DC. The U.S. is easily falling behind on a lot of this stuff. It's really important that that the U.S. I mean, with foundations like Kavli, with Carnegie investing in our resources, but also the federal government has to invest in this stuff. And if it weren't for that, I bet we could ask you, Bob, how much would you have accomplished?
It's absolutely critical. Yeah. I mean, I think any professor, I mean, everybody spends a lot of time trying to raise money. Yeah. I mean, that's. Yeah. And I would add that even when you have these small companies that, do these things, they spend a lot of time raising money, and the road is long. A lot of the time you fail, more than you succeed, whether it's in academia or the tiny companies. I mean, it's just it's hard to raise money.
Yeah, it's hard. So, okay, let's thank Professor Langer again for an amazing talk. And just being a really nice guy, because as you say, in science, that's, not a lot of nice people, but not everybody. And then also to Professor Frank Sesno for just being able to pull everything out. And I thank you, the audience, for coming. Pay attention. We have many more of these coming in the future. And thank you to Cynthia Friend and Kavli for being so exciting and providing so much resources for research, but also for these kinds of events. Thank you. Good night, everybody.